Good afternoon, everyone, and thank you for being connected today during this challenging time. My talk today is about selecting the right technologies to advancing the manufacturing processes to the required level of productivity to meet the current and future demand for gene therapies. Repligen has a concerted initiative on gene therapy to partner with the industry to bring the technology-ready solutions to accelerate the journey from gene to therapy. The manufacturing challenges to make gene therapy a global success are well identified. We need to increase the upstream and downstream productivity to produce enough doses to meet the demand, and this requires a thorough process optimization using scalable techniques, and we need to do it quickly to reduce the time to market using high throughput techniques. And we cannot compromise with the product safety and quality. And we can achieve that thanks to the adoption of automated systems and closed processes. So let's consider these issues and solutions one after the other. The current process used to produce viral vectors, let's call it gene therapy manufacturing 1.0, is often limited in productivity and scalability from the use of adherent cell culture and transient expression, and also results in low downstream yield. Ongoing developments related to cell lines, media, and process optimization will lead us, hopefully within the, let's say, next 2-3 years, to the gene therapy manufacturing 2.0 with producer cell lines grown in suspension mode at high cell density to produce high virus titers, and optimized purification processes to prevent virus loss or inactivation. So it's not surprising to see almost all gene therapy companies now adapting suspension cell culture for the development of their new treatments. This data here is just one example published about a successful case of AAV vectors produced from HEK293 cells adapted to suspension and showing an increased overall productivity coming from the suspension cell culture. The transition from adherent to suspension adds safety to the process as no animal-derived material is needed, and also significantly facilitates the scale-up of the process. But the titers in suspension mode are generally lower compared to that of obtaining adherent mode. So even if the transition to suspension is beneficial for safety and scalability reasons, some further improvement is required to increase the titer, and this can be achieved by intensifying the process, going from batch to perfusion cell culture. Here is such an example coming from one of our customers using the Repligen XL-ATF technology, which is a cell retention technique enabling very high cell density in perfusion mode, and which is already broadly adopted for the production of recombinant proteins and mAbs. But it's also implemented now on gene therapy processes with already good success. Here, the productivity of optimized XL-ATF perfusion runs was compared to that of batch cell culture. The XL-ATF perfusion process enabled a higher cell density at transfection, and this, combined with optimization of cell culture, led to a higher than 100-fold capsid productivity increase. So I believe this demonstrates that we are ready, technology ready, to overcome the upstream productivity challenge thanks to the adoption of suspension cell culture and perfusion processes. Can we go even further? Yes, using the same tools, but now with producer cell lines that are currently being developed. So now let's look at the challenges on the downstream side of the process. We see a cumulative loss effect during the downstream purification of viral vectors, lowering the yield step after step, sometimes resulting in the loss of about 90% of the active product. Most of the loss, as you can see, is coming from the two common chromatography steps with non-optimized separation, leading to compromise between purity and yield, but also from harsh conditions used in terms of pH conductivity, leading to virus inactivation. Additionally, non-optimized TFF steps will also lead to significant product losses due to shear forces, the product being bound to the membrane, or simply loss in the older volume because of wrong sizing. So it's therefore critical to use the right technology during the process development to optimize this process. On the chromatography side, the use of small column high throughput screening tools will help you rapidly identifying the best separation conditions among a huge number of combinations between resin types, chemistries, flow rates, capacity, etc. And the identified process can then rapidly be scaled up using the corresponding scale-up technology. At Repligen, we offer such a complete tool set called Opus Pre-packed columns, with a comprehensive choice of chromatography resins and chemistries pre-packed in different formats and scales. Starting with the RoboColumns, used for rapid and high throughput screening of separation conditions, before fine-tuning these conditions to a scalable process with the mini-chrome columns, then validate the process with the Valichrome columns, and finally scaling up to the process scale. Thanks to these tools, you really optimize and scale up your chromatography process steps. By the way, you will get more details about this specific topic during this talk at 3:30 this afternoon. So, after having done the chromatography part, let's improve the TFF performance. The first thing we need to consider is selecting the best format between hollow fibers and flat sheet cassettes, which will generate more or less shear-related stress. If your product is sensitive to shear, like envelope lentiviruses, the hollow fiber is better suited. But for less sensitive vectors like AAV, the TangenX flat sheet cassettes provide a great option. So this graph here clearly demonstrates the impact of the higher shear generated with the TFF cassettes compared to hollow fibers, with only 50% of the lentiviral infectious particles recovered compared to full recovery with hollow fibers. This is actually a result that we see quite often for lentivirus applications. So after having selected the TFF format, here it's the hollow fibers, but again, it could be flat sheet cassettes for AAV. So having selected the format, it's important to evaluate multiple molecular weight cutoff membranes to first isolate the first with the best yield, but also contributing to the purification by removing impurities. In this case here, we do not see a clear difference in terms of recovery of infectious particles. But the highest molecular weight cutoff showed better HCP removal and was therefore selected. Finally, some flux optimization will decide the process conditions. And very importantly, from there, we will guide you step by step to define your process scale-up with filter sizing, operating conditions, and selecting the automated hardware corresponding to each scale to run your process under optimum conditions with minimum human intervention to save on handling time, but also minimize the risk of human error. That brings us to the last challenge to address for gene therapy manufacturing, which is securing the product quality and safety by implementing closed processing. We can definitely help securing the process by providing you complete and integrated single-use flow path for each unit operation. Even further, we want to give you the opportunity to get the flow path customized to your own process, delivered pre-assembled, gamma irradiated, and ready to connect via sterile connections, making a further strong step towards safe processing for gene therapy manufacturing. So starting with the gene therapy manufacturing 1.0, going through technology-driven process optimization and innovation, we are now coming closer to that gene therapy manufacturing 2.0, where the viral vectors will be continuously produced by producer cells using intensified suspension cell culture run in perfusion mode and purified through a secured and high efficiency downstream process. With this, I thank you for your attention, and hopefully I would have a few more minutes left to take any question. But anyway, feel free to reach out to me. Here is my email if you need further information about our technologies for gene therapy. Thanks again.